Hello my dear students, this is Dr. 𝗵𝗺𝗺, your biochemistry faculty. Well, in this video we are going to discuss about the hematopoietic vitamins. Now what are hematopoietic vitamins, why they are known as hematopoietic, and in which class of vitamins they fall, what are their functions, what are their active forms, these all we are going to study now. So let's start up with hematopoietic vitamins. Now see, as it is clear from the word itself, hemato means something related to the blood. So we can say that this class of vitamins is going to include those which are related to the formation of the blood, obviously. So hematopoietic vitamins are B9 and B12. Now coming over to this belongs to B complex vitamins because B is there, so they will come in the category of B complex vitamins. And whenever we talk about B complex, they are water soluble. So I can simply say that they are the water soluble vitamins. So if we talk about their, I would say ki synthesis and all, they are, since they are soluble in water, once their function is over, they will be excreted in urine as such, right? So they cannot be stored. Now coming over to this, first of all, we are going to take up this vitamin B9, which is also known as folate or folic acid. And this folate is actually derived from the word folinum. And this folinum means green vegetables or green leafy vegetables will come under this. So I can say that folic acid or B9 is rich in green leafy vegetables. And if we talk about the structure of folic acid, it has folacin in it. It has a pteroic acid or pteridine present in this. Now coming over to this, what is the active form of this folic acid? Now always remember, this folic acid is required for 1-carbon metabolism. Very, very important. It gives rise to 1-carbon metabolism. Now what is this 1-carbon metabolism which gives 1 carbon? Obviously. Now how this 1 carbon is given to the body? Now this 1 carbon can be present in the form of methyl group, CH3. It can be present in the form of methylene group, CH2. It can be present in the form of methynyl group, CH, or it can be present in the form of formyl group, which is HCHO, or it is in the form of formimino group. Everywhere, if we see, there is one thing which is common, which is C. Everywhere. Right, that means from where this 1 carbon is coming. So this 1 carbon is coming from folic acid. And if we talk about the active forms of this folic acid is active in as THFA, which stands for Tetrahydrofolate. So if it is carrying any particular group, it will be categorized like that, like N5-methyl THF, N5-methylene THF, methynyl THF, and so on. So it's like wherever the position, wherever it is present, it will be named like that. But always remember, the 1-carbon metabolism, folic acid is the most important vitamin, which is actually a hematopoietic vitamin, is required. Now coming over to this, that first of all, what are the functions? what is the source of this 1-methyl group. So if we talk about 1-methyl group, or you can say C group. So what happens here is this methylene group, this methylene will be required for the synthesis of thymine, or you can say DNA synthesis. And this reaction needs to be strictly irreversible. This is a very important reaction. I will discuss this reaction in detail also, but here I am just writing that this methylene, basically it will be required for the synthesis of thymine monophosphate, which is the base present in DNA. Then this methylene can be converted into methynyl group. Okay, it can be converted into methyl group, and this methyl group is required for the conversion of homocysteine to methionine, taking B12 as a coenzyme. This reaction is a very important reaction. And from where this is coming? It is coming from, simply you can say that serine is converted into glycine, or I can say that serine, glycine, like this, third carbon to second carbon, and this methylene, glycine to serine. Right? And then we can say that there is one more addition of a formyl group also, because whenever they are giving, you can see that if we talk about methyl is CH3, methylene is CH2, methynyl is CH, so they are giving one more hydrogen at the same time. So it can be converted back also. Methylene can be converted into, methynyl can be converted into methylene, like that. Similarly, this formyl group, where it is written, it is required for the synthesis of purines. So I can say that methynyl and then formyl group for purine synthesis. Right? So this is how you can say it is utilized. Now coming over to this, what are the biochemical functions of this folic acid? Now the most important biochemical functions. The most important function is conversion of uracil to thymine. Conversion of uracil to thymine. Now, we all know that uracil is the base present in RNA and thymine is the base present in DNA. And the first pyrimidine which is synthesized is uracil. So from uracil, because in our body DNA is also there, DNA, RNA both are synthesized, the nucleotides. So this uracil will be converted into thymine by folic acid or folate. Alright, so the reaction is simply, the name of the enzyme is folate reductase. And it will give a methylene group, and this is the only reaction which is irreversible. Means I can say that this methylene will be given and this is a irreversible reaction. Reversible reaction. and all the anti-cancerous drugs are inhibitors of this. So I can say that anti-cancerous drug like methotrexate is an inhibitor of this enzyme folate reductase, right. So this is one reaction, conversion of uracil to thymine. Then if we talk about the second reaction is a very, very important reaction in which homocysteine is converted into methionine. So if we see the structure of homocysteine and methionine, the only difference is of the methyl group. That means if we put methyl group in homocysteine, it will be converted to methionine. So what happens here is that the donor here is folic acid, but the carrier is B12 here. So the donor here is folate and the carrier here is B12. And the name of the enzyme is methionine synthase. Right. So what happens here is, you can simply say that methyl B12 will be converted back to B12 and methyl THF will be converted into THF like this. So I can say that this folic acid is the one which is giving methyl group, matlab handing over the methyl group to B12 and which will give it to methionine synthase forming methionine. This is a very, very important reaction of folic acid. So these two are very important reactions, and along with this other reaction can be, we can say glycine to serine. So basically the enzyme here is hydroxy methyl transferase. But here the main one is folate. Then if we talk about the fourth reaction, formation of purines. So if we talk about this C2 and C8 of purine, they come from folic acid because purines are synthesized by a de novo pathway. So this is the reaction basically. So it is 1, 2, 3, 4, 5, 6, 7, 8 and 9. So this and this, they will come from folic acid. So this is another very, very important function of folic acid. Then the most important, again one more important function of folic acid is it plays a very, very important role in the metabolism of histidine. So what role it has to play in the metabolism of histidine? Metabolism of histidine. We all know histidine is a very important, I would say essential amino acid which is basic in nature also. So this histidine, when it undergoes degradation, it forms urocanate, and this urocanate will be converted into FIGLU. FIGLU, which stands for formimino glutamate. Right. So what will happen here is this FIGLU in the presence of B9 will give this formimino group, right, and it will be converted back to glutamate. Right, glutamate which can further form glucose or glucogenic. Right. So what will happen? We say that if B9 is not there, there is accumulation of FIGLU in urine. Right, this is the most important function of vitamin B9. Okay, so these are the important reactions catalyzed by B9. Then coming over to, again after this, what we are studying is, we are studying the biochemical manifestations or the deficiency manifestations. Now, the most important deficiency manifestation of B9 is folate trap. This is very important. There is defective DNA synthesis. Right, then folic acid deficiency leads to neural tube defects also, because in pregnancy this folic acid is mandatory, it is very essential for the fetus. So neural tube defects like 𝗵𝗵 bifida is there. Since there is defect in DNA synthesis, it will lead to megaloblastic anemia. Right. So these are the four very important deficiency manifestations of folic acid. Now the question is, first of all, what do you mean by this term folate trap? Okay. So what happens here is, because this is the only reaction in our body which requires folic acid and B12, and the reaction is this: homocysteine forming methionine. So what happens here is, B12 is just acting as a carrier, so I can simply write CH3B12. And this is coming from CH3THF. Now, so there can be two cases. If B9 is deficient or B12 is deficient. Okay, so if we talk, we will take the cases separately. If I say B12 is deficient, right? B12 is deficient. Now B9 does not know that B12 is deficient. So what will happen is, B9 will come and B9 will keep on donating its methyl group. A stage will come when the entire folic acid will be trapped here. Now the question is, if folic acid is trapped here, other reactions which still require carbon, which still require methyl group from folic acid, they are going to suffer. Okay, so this term is known as folate trap, and folate trap is mainly seen in B12 deficiency. So what will happen is, in B12 deficiency we can say that ki B9 will donate the methyl group, right? So it will not be available for rest of the reactions. So it will not be available for the other reactions. So it will lead to the trapment of folic acid, it will lead to megaloblastic anemia, it will lead to, in fact, all the things which are related to B9 deficiency. Why? Because folic acid ka carbon, folic acid ka methyl group is trapped, and it will also lead to homocysteinuria. So homocysteinuria can be of two types here. We can simply say that this homocysteinuria right, classic and non-classic. So if we have to differentiate between classic and, because if we talk about the biochemical investigation, this cyanide nitroprusside test will be positive in all the homocysteinurias, but the basic difference is, in classic, deficiency of B6 is there, In non-classic, B9 or B12 is there. Okay. Then in B6 deficiency, if we talk about methionine, methionine is normal, whereas cysteine is reduced. And if we talk about B9, here methionine and cysteine will be normal. So we can say that homocysteinuria non-classic is due to the deficiency of either B9 or B12, but in B12 deficiency it will lead to folate trapment of folic acid, right. So these two are the very important, I would say, the deficiency manifestations with the reactions. We can say there will be trapment of folic acid. Why there will be a defect in DNA synthesis? Why? Because uracil will not be converted into thymine. Then it will lead to neural tube defects leading to 𝗵𝗵𝗵𝗵, and then megaloblastic anemia will be there. So these four are the very important deficiency manifestations of folic acid. And then next we come over to the assessment. How do we assess? So if we talk about the biochemical assessment, the first one is again homocysteinuria. Homocysteinuria or homocysteine in urine. In the urine and blood. Second important one is peripheral smear. Right. Third important one is, which is the most important one, I would say, histidine load test. Histidine load test, or which is commonly known as Figlu excretion. Very important. Figlu excretion. This is the most important test to check the presence of check the deficiency of B9 in a patient. To eat. So obviously, what is the reaction behind that? This histidine forms urocanate and then it forms Figlu. And this Figlu in the presence of B9 will be converted into glutamate. So if B9 is deficient in that patient, so automatically the concentration of Figlu will increase and this Figlu will be excreted in urine, which indicates B9 deficiency. And one very important thing, because B9 and B12 are catalyzing the reaction there. I told you there is one common reaction between B9 and B12. So here, in case if B9 is deficient, methyl-malonate levels will be normal. Methyl-malonate levels will be normal in B9 deficiency. Right? And then the biochemical assessment is done by ICAR test. Amino, imidazole, carboxamide, ribosyl 5-phosphate. Right? So these are the tests by which we can detect the deficiency of B9, but the most frequently used test is histidine load test. So this is a very important vitamin which is hematopoietic in nature. Then another important thing is कि whenever this folic acid is deficient, it might lead to, but there are so many things there. The epigenetic modification will also be hampered because again the source of methyl group is this. So always keep one thing in mind whenever the concept of one carbon metabolism comes. You always have to go for folic acid whose active form is tetrahydrofolate, which is carrying this either methyl group or methinyl group, or you can say methyl group, methylene group for amino group, or it is carrying formyl group. So ultimately it is coming from there. And another important thing is the source is conversion of glycine to serine and vice versa. Because always remember glycine and serine both are non-essential amino acids and both are synthesized by a reversible reaction interdependent on one another. And always keep one thing in mind, this folate is actually present in the green leafy vegetables. So if we cook green leafy vegetables for a longer time, so what will happen is the folic acid that efficacy will be destroyed. So we should avoid, I will just write it also, that avoid heating green leafy vegetables for a longer time. Why? Because cooking destroys, or excessive heat destroys folic acid. This is very important, right? So this is all about folic acid, which is a very, very important hematopoietic vitamin. Thank you so much.